Hello, everyone. This is Alex Puschel, and I will be discussing a technique that plays a central role in my first rotations project. So without further ado, let's get into it. I'll be discussing leveraging genetic engineering to produce useful protein constructs. So an overview is I will discuss what heterologous protein expression is, its benefits and use cases, challenges to implementation. The other aspect is the plasmid expression system LEMO 21 DE3 system, a tunable two plasmid system for expressing your gene of interest. At right, we have the AlphaFold predictive model of insulin, one of probably the most recognizable recombinantly expressed proteins, and that's expressed in E. coli. Specifically for this, I'd like to focus mainly on E. coli, but heterologous protein expression can be accomplished in a number of cell lines, such as mammalian cells, bacteria, or fungi. What is heterologous protein expression? Expression of a gene or gene fragment in a host organism that does not naturally express that gene or gene fragment. So specifically, since we're focusing on E. coli, it's a well-established model organism, has rapid growth kinetics, and when you're purifying, you don't really need a large volume of animal or plant tissue like you would need in the past to even simply isolate insulin. Plug and play protein motifs allow for easy construction, and you can even incorporate non-canonical amino acids or have proteins with specific characteristics. At right, we have a typical E. coli vector with its selectable marker, so you make sure you get the cells that only express your plasmid, and a number of genetic pieces that go with it, such as a promoter or a ribosomal binding site. There's a number of challenges to heterologous protein expression as well. Specifically, organism selection is key, specifically focusing on E. coli here. But if you need something like that, a protein that has a lot of post-translational modifications, you can't do that in bacteria. You might need a mammalian cell or something else eukaryotic. You might be trying to express a protein that's either inherently toxic or as the amount of construct you make increases, it becomes toxic over time. And, increasing protein construct concentration, which is something we want, can lead to toxicity. Membrane proteins tend to be tricky. If they're not transported to the membrane fast enough to keep up with expression, then you end up with inclusion bodies. There's codon biases, so the pool of tRNAs that you're drawing from, the codon that you selected might have a very low pool. Post-translational modifications, like I said, you may need to pick something more eukaryotic, and E. coli tend to not tolerate large proteins very well. Next, we have the LEMO 21 DE3 system, which I said earlier is a two plasmid tunable system, and it's really good for those tricky proteins, toxic, solubility issues, or ends up needing to be membrane bound. It has a highly titratable rhamnose promoter, which makes it super easy to tune your expression levels. And because it only has that one really good tuning mechanism, optimization is easy. So at right, we have the genetic schematic. This is almost constitutively produced T7 RNA polymerase, and that goes and strongly binds this promoter and leads to high expression of your construct. And then you tune it by titrating this rhamnose, which goes and binds this promoter and makes T7 lysase and this lysozyme. And this allows you to modulate dampening of production, since this will inhibit this and start to modulate this production. And that'll lead to dampening or increasing of your gene of interest construct. Next, we have the proof of concept. So how is this going to look? On the graph at left, this is actually counterintuitive to a typical inhibition mediated expression mechanism. But as you express the T7 polymerase at low concentration, as it's induced, you'll actually see you get low expression because you're expressing so highly that you're leading to toxicity rather quickly. But as you see, as you titrate in rhamnose, you get some dampening effect, and that starts to increase the amount of protein that you're actually making. And that leads to an optimal point, and then you're dampening too much. At right, we have the expression of LEMO 21 versus another OK protein expression model. And you can see these three are bacterial protein membrane, bacterial protein membrane proteins, and you still get really high expression with them. And then these are human, get really high expression, and then these are just soluble protein. And this shows that LEMO is very good at expressing heterologous proteins. Thank you. And here is my references.